Good afternoon, everyone. Welcome back to the TBL number 4. The submodule is going to cover the types of skeletal muscle fibers, and this is Lin. Skeletal muscle fibers that compose a muscle are differentiated into three categories based on these main two criteria, which is the ability for it to generate a contraction and the primary means that it utilizes ATP. The power is related to the diameter of the muscle fiber. The speed and duration is related to the ability for the myosin ATPase to hydrolyze ATP. It also is relevant to the quickness of the actual potential propagation, as well as how quickly the calcium is released and reabsorbed. When we consider the supply for ATP, there's two major pathways, the oxidative versus glycolytic. Oxidative fibers means that they utilize oxygen as a final electron acceptor, and they can enable aerobic cellular respiration. These ones are fatigue resistance. An appearance on cells will look more reddish in fiber, and they're supplied by an extensive amount of capillaries. You'll also find numerous mitochondria organelles in these cells. Glycolytic fibers are white in coloration, and they predominantly use glycolysis. There's a very limited amount of capillary supply and a much fewer amount of mitochondria and myoglobin. Because of this, they are more fatigable. So let's take a look at the three types of fibers. The first kind of fiber is called slow oxidative. We abbreviate that as SO fibers, aka type 1. They have small diameters, they contain slow myosin ATPase, and they have large amounts of myoglobin and mitochondria, producing a much slower, less powerful contractions, and on a stain, they tend to look more darker reddish in color. On the right-hand side, you can see a graph showing you the slow oxidative fibers with the x-axis showing you time, expressed in minutes, and then the y-axis of tension expressed in milligrams. The second type of the muscle fiber are called fast oxidative, FO for short. These are also known as 2A, which is intermediate fibers. They can generate contractions that are fast and powerful. Their vascular supply is a little bit less extensive, so that oxygen and nutrient delivery is slower, and there are fewer mitochondria. They will appear in a light reddish tint and are the least numerous type in the body. Over here's a picture showing you the fast oxidative glycolytic fibers, FO. And as you can see, endurance goes down after a certain amount of time. About 5 to 6 minutes in, the endurance drops down. The final type of the muscle fiber are called fast glycolytic, also known as FG fibers. These ones are fast anaerobic fibers, also known as 2X. They contain fast myosin ATPase with extremely large diameter. They're fewer mitochondria, they lack myoglobin, and produce both a powerful and speedy contraction. They're used mostly for short bursts, like a max rep. They rely solely on glycolysis, and the coloration is going to be white due to a lack of the myoglobin protein. You can see right here, after under 2 minutes, there's a substantial drop in the tension that these fibers can generate. So comparing the same graphs of time on the x-axis, you can see these two SO and FO fibers are able to sustain contraction at a lot longer time, over 8 minutes, whereas for fast glycolytic, it's going to be really less than 1 minute or 2. Okay, this next submodule is going to be on smooth muscle. Smooth muscle is located throughout the body, typically about 2% of the entire body weight of an adult. They're located inside vessels of the cardiovascular system. They're found in the bronchioles of the respiratory system, found inside the stomach, the small intestine, large intestines, the ureter of the urinary system, the uterus of the female reproductive system, and of course the iris and the erector pili muscles. There are two types of smooth muscle. They are broken down into the multi-unit smooth muscle and the single-unit smooth muscle. The multi-unit smooth muscle contracts individually, whereas a single-unit smooth muscle contracts as a synctium or is unison. These ones are also more common. Because they are able to contract synchronously, they are linked together via gap junctions. They're located predominantly in your GI tract, your bile ducts, your ureters, your uterus, and many, many blood vessels. Whereas the multi-unit receive contraction individually, and they're found in your iris ciliary muscles, auricular pili muscles, larger air passageways in the respiratory system, as well as large arteries. On the right-hand side, you can see a visual differentiation between the two. Letter A shows you a multi-unit. You can see they're a little bit more scattered and sparse. Individual nucleuses shown inside the cell. On the right-hand side, we have the unitary smooth muscle, where they're all lumped together, and it's lining the endothelium so that these cells are stimulated at one time altogether. Smooth muscle cells are small, with a diameter of approximately 5 to 10 micrometers. And the length of these cells can range from 20 to about 200 micrometers. The sarcolemma contains multiple types of calcium ion voltage-gated channels and chemically gated channels. There are no T-tubules present in these cells, but instead, they have these uniquely modified invaginations called calveoli. These small invaginations increase the surface area to allow calcium to excite different parts of the cell. The sarcoplasmic reticulum is also sparse. It doesn't contain any troponin complex, but instead contains a calcium binding protein called calmodulin. This is expressed as CAM protein for short. It also lacks the sarcomeres and Z-discs, therefore you do not see the striations in a microscopic view. The cytoskeletal component is composed of extensive intermediate filaments, and they're anchored by these dense bodies where the actin filaments can dock, and dense plaques where the intermediate filaments can attach to the inner compartments of the sarcolemma. Over here on the right, you can see the dense plaques. They're the structures that align to the cell membrane, and the dense bodies which will anchor and hold on to the actin thin filaments. The smooth muscle filaments have actin myosin, and there's a typo right here. It does not have troponin. They elicit a long, slow contraction, and this is a result of a long latent period because of a slower ATP's activity. As a result, they're very fatigue resistant. There's a very low energy requirement compared to skeletal muscle, and they engage in a mechanism known as latch bridge. Because of their lack of the Z-discs, there really is no limitation to that length to tension curve. They can sustain a contraction and extend it for a much longer period of time. You do see the myosin heads in the thick filaments. Over on the right-hand side is a snapshot of a smooth muscle. You can see the nuclei. There's no striations at all, but they're scattered sparsely throughout. Nucleus is over here, nucleus over here, nucleus over here, and another one over here. Again, the cartoon shows you that when the smooth muscle is relaxed, it is elongated upwards, and when it's contract, it has more of a twisting motion rather than a sliding motion. Let's talk a little bit about the mechanisms of contraction. The smooth muscle contraction is initiated by calcium ions. This requires ATP and involves the thin filaments sliding, but as a result, twisting on the thick filaments. It begins by the stimulus opening up a voltage-gated calcium channel. Calcium then rushes into the sarcoplasm, primarily from the interstitial fluid on the outside. Calcium will then bind to the calmodulin protein, a receiver, and form a calcium-calmodulin complex. The calcium-calmodulin complex will then activate an enzyme, myosin light chain kinase, also known as MLCK. MLCK's job is to phosphorylate the myosin head, activating the myosin, which is a slow process. The activated myosin head can then bind onto actin and begin that cross-bridge sliding, twisting motion. Recall that the myosin ATPase hydrolyzes ATP, which provides the energy for that power stroke, and this process is repeated. The force is generated, is transferred to anchoring filaments, and then results in the smooth muscle contraction. Over here on the right is a picture showing you that. So as the cytoskeletal calcium increases, the inactivated calmodulin is activated. That will then trigger the activation of the inactivated MLCK to turn on, which will then phosphorylate the myosin head. that allows the cross bridge to begin to form. This process is repeated, and eventually the smooth muscle is able to contract. Relaxation, on the other side, requires the cessation of this process. So you begin by removing the stimulus, you remove the calcium, and then you dephosphorylate the myosin head with another enzyme. This enzyme that counteracts this is called myosin light chain phosphatase, MLCP. This will then remove the phosphate from the myosin head to remove the cross bridge, and this is a result of as calcium begins to drop. Smooth muscles generate an action potential that is spontaneous. On the right-hand side is a visual showing you the slow wave potentials that is found in the unitary smooth muscle. And these can eventually lead to what we call spontaneous action potential. These cells are self-excitatory. They don't require input from the nervous system, as the cell membranes have a lot more VGCs for calcium channels, but a lot fewer for the voltage-gated sodium channels. You can see that it looks kind of like a spike. The spike potential represents this spontaneous depolarization to the threshold resulting from stretching. So when you stretch the smooth muscle, it will self-trigger an action potential that will then send a signal. On the upper left-hand side in letter A, you can see this is a typical smooth muscle action potential that is elicited via an external stimulus. Over the course of time, this slow wave potential can also be generated as a rhythmic set of electrical waves that occurs simply from stretching and contraction. And over on the bottom, letter C shows you that action potentials do have a plateau, which is shown in different parts. Like in the uterus, there's a plateau phase where it extends on as a result of calcium channels dragging on this duration of the signal. As a result, sequential signals, we have slow waves, also known as pacemaker waves. These are drifts up and down due to regular variations of ion fluxing across the membrane. The specific details of why this is occurring, we do not know, but most of the evidence suggests that there's natural movement of ions when you stretch, and as a result, you can generate this slow wave potential. Controlling and regulation of smooth muscle. So smooth muscle contraction and relaxation is not controlled voluntarily, but rather controlled by the autonomic nervous system in response to specific neurotransmitters and types of receptors that the neurotransmitters bind to. Half of all the smooth muscle is likely to be initiated by stimulatory factors directly released onto smooth muscle. So either acetylcholine and norepinephrine, those chemicals dictate whether or not the smooth muscle will constrict or to dilate. You will also notice a structure called a diffuse junction. These are receptors that are located across entire sarcolemma, and as a result, the stimulation can spread from cell to cell via gap junctions. As a result, you get the synchronized contraction as a whole unit. Over here on the right, you can see innervation of a smooth muscle by autonomic nervous branches, and these ones secrete these chemicals across, and then the receptors are located all over the cell. Small gap junctions will then be able to synchronize the constriction or dilation. On the bottom, the multi-unit smooth muscle, each cell is stimulated independently by a specific neurotransmitter. But they are closely linked to these little pockets called varicosities. These are where neurotransmitters are stored, and you may have some areas that are excited while some areas are inhibited. Contact junctions are found inside these multi-unit varicosities and are close to the muscle membrane, roughly about 20 to 30 nanometers. And there's two mechanisms that control this. There's the myogenic response, where contraction is a result of stretching, as well as the stress-relax response, which is natural relaxation after you overstretch the muscle. The stress-relax response occurs when smooth muscle is stressed by being stretched for extended periods of time, eventually pushing into the relaxed state. Smooth muscles can also respond to various hormones, a drop in pH, low amounts of oxygen concentration, an increased amount of CO2 levels, and of course certain drugs and pacemaker cells.